So resending the computer. Here we go.
This one? Or is it that one? Maybe not. Did I break it?
It needs to be on VJ.
Yeah.
There you go. This is what it looks like on the other end.
Cool.
Without the stuff.
Great.
Alright, got an hour and 20 minutes.
Perfect.
To get through.
It's weather.
What?
The weather.
Oh,
It's
yeah.
making it extra bad.
Yeah, we're home.
Should have just figured it out. I'm trying to figure out how to do the gas exchange.
Oh, yeah.
Brian got it, and I'm like, I don't know how to do this.
I'm really liking the fact that I took that, like, miniature literary or literature review or whatever.
Well, he automatically started talking about it.
Yeah.
How's it going? Thanks for meeting, Pastor.
Oh, yeah.
Do you guys actually know each other after all? Oh, no. I think I might like this word, actually.
Yeah, no, you got
We
it.
both like.
That makes sense. Go for that.
We both like words on your
Yeah,
website.
been over in the first collection.
Oh,
quite some time. I'm willing to bet that.
How big is your, I don't even know what
It's a gas analyzer.
That's
So it is like this big. It's how to keep it.
Where's the cool kid sitting in here? I came to feel it the first time in the Usually
goes somewhere where you walk them.
Very elementary.
I don't think I'm nervous here.
I'm German.
Yeah, right. Pretty anyway.
I'm very telling me.
Yes, so it's funny. Just your back.
Hello.
I think we quickly, before you did the GIS, did you GIS?
Am I in GIS?
No, I was TAing GIS. I think it
Oh,
was
yeah, that was me. Yeah.
It's like,
Let's
I
start
think
with
there's a few open computers. Hear my dumb voice.
Thank you.
That, like, what's it, emergency and stuff or whatever?
How's it going?
Good.
I like that.
Facing down Brandon's lab.
Yeah, it is.
I thought it did. I think for me.
I know. I sometimes feel like I do.
If I drink gas and stuff, I'll just be okay.
Everything I do is alright. I mean, more or less. My problem is I'm just a little too chatty about totally low alcohol, which means literally everyone in the car. Oh yeah, I mean, I think everyone does. Like, if what I've ever talked about.
Back to be on your back.
It was tempting. And then like,
Yeah.
I tried
I didn't do it the first day. I tried to do it yesterday.
Probably smart.
and I can't remember.
Yeah, me too.
Yeah, probably.
Yeah, I went to breakouts with a lot of people all the time.
Oh, for like
For like school back class, I'm like, I need not that uses.
that's program.
Passion forever is.
But then you also get
Sorry. Thank you.
Like, go back.
just like
Legally. I got all my stuff done this morning.
I think it's like
that.
that.
It's another file. Like,
Grinding folks in a
And then like force it to do what I need to do. So rather than trying to get them, I'm like, all this shit.
They probably
It's a different weekend.
Those functions are always going to be done. Like hacking is
Run it,
so
I don't know.
So I told her there's something where if you run a function, it's depending on the So my function,
like somewhere in the hallway or something, like 1 minute after.
you just memorize like I just memorize how many rows down.
Yeah.
I don't know what those
Reading the columns and stuff. Like, it was definitely like yeah, so like my functions, like, I've written them all down. They're more
I didn't know you I knocked it, you know, I didn't know that everybody did it. I had a meeting today,
pencils around the face. So I presented it that much earlier. No. Do you have, were you getting
I know I have to go down there and work all along. We're mostly focusing on
Thanks everyone for coming to seminar and for allowing me to infiltrate it once again. So graduate students, you have impeccable taste in nominating Dr. McCoy to join us today. He's visiting from Eastern Carolina University, and I've always had a really difficult time describing his body of work. It is really, really diverse. He's a quantitative ecologist. He works in a whole bunch of different systems. There's species interaction and a lot of predator-prey ecology and aquatic taxa and herbs and insects and more. And also, it should be noted that he was on the team that produced the best paper that's ever been written. It's really cool. I think you're going to see a slide about it or something.
Yeah.
It's about how fish support pollination services. And today he's going to be talking about something that I find really awesome, which is predator diversity and trophic cascades. And we will learn about that right now. Welcome. Thank you so much for joining us.
Thank you. Can everybody hear me okay?
Oh, wait. No, hold on. You can't start yet.
Okay.
We're moving to Nick's afterwards. We have the big booth reserved there, so please join us. Mike is really cool and great to pound brews with.
It's my favorite thing, actually. Yeah, so you can hear me okay? Okay, yeah, thanks for the invitation. I'm really looking forward to telling you guys about sort of 3 pieces of my work, maybe 4, depending on how much time I have, that's really focused on sort of improving predator-prey ecology theory in terms of how we approach multiple predator systems in particular. So my interest in sort of predator-prey ecology really started as a young graduate student when I read this classic paper by Hairston, Smith, and Slobodkin. And one of the things I really liked about this paper wasn't necessarily just the content, but it was the approach. So this is kind of a pithy thought experiment where they use sort of deductive reasoning to logically construct an argument to explain why are there so many plants on the earth when there's so many herbivores around. And through the sort of simple chain of logic, they deduced that you can have the world can be green because there are predators controlling herbivore populations. Now that's an oversimplification, but that basically laid out the logic for all of modern food web theory. And there's been lots of confirmation of their basic logic. For example, we know classic examples like the introduction of wolves in the Yellowstone, changing the structure of the vegetation communities and lots of other things there by changing the behavior and the abundance of prey. Similar sort of classic example is the sea otters off the coast of California. By eating urchins can stop kelp forests from being converted into mudflats. And in some work I did earlier in my career that Carmen referred to, sometimes the effects of predators can be cascade to impact things that are quite far away that you might not expect. For example, predatory fish, by eating larval dragonflies, can indirectly affect the pollination success of plants growing nearby ponds through an indirect cascade of reducing the number of larval dragonflies, reduces the number of large adult dragonflies, which enhances the number of pollinators visiting plants around ponds. And so this sort of was my early interest in predators, but it sort of continued because we know that predator communities are changing and probably at an increasing rate. And this is due both to invasions and extinctions. And most cases of introductions of animals, at least, actually end up being predatory animals. And most cases of extinctions are large predators. And sometimes these introductions and extinctions of predators lead to huge ecological and sometimes socioeconomic implications. So what I want to do today is tell you 3, and there's kind of 2 stories embedded in this second one, sort of scenarios of predation where I've hopefully contributed a little bit of additional knowledge. And the first of these is talking about predation pulses, or in other words, asking how do temporal pulses and top-down forces affect prey populations and ecological processes. And this question sort of stemmed out of the fact that there's been lots and lots of research focused on how resource pulses can cascade through food webs and change dynamics of ecosystems. For example, large seed masts and acorns, or the bodies of cicadas, periodical cicadas, after mass emergences fall to the ground and sort of fertilize ecosystems, or a whale carcass washing up on the beach, or the transfer of nutrients from agricultural fields to ponds by geese. All of these things have been shown to have big effects on communities. But all of this work was focused from the bottom up, and very little of it looking at processes happening from the top down. But we know that top-down pulses are also common. For example, some things I worked on in consumer aggregations, like periwinkle snails in salt marshes, or crown of thorns sea stars on coral reefs. So you get these huge aggregations of consumers. Wildebeest on the African savanna is another example of snails, but you can think of lots of examples of aggregations, locust or another. But then there's also lots of opportunistic, transient, and stochastic predators, like a heron visiting a small wetland pond, or a little boy in a rock pool. Right? These are transient, stochastic predation events that can change the structure and dynamics of these communities in a different way than sort of constant, consistent predation. And then we can also have episodic and cyclic predation scenarios, like fisheries seasons for harvesting, or fish visiting cleaner shrimp on a coral reef. Or one of my favorite examples is the mass emergence. So if you notice all this dark matter on the side of this pond here, all of that are metamorphosing frogs emerging from this pond at the same time. So millions of little frogs coming out and eating organisms in the terrestrial landscape. So you can get these pulsatile and sometimes stochastic pulses of top-down consumer pressure, but there's almost no theory addressing these kinds of issues, with the exception of a couple of really obscure papers, one showing that pulse vaccination strategies can actually lead to more rapid eradication of, in this case, measles, or another scenario looking at how pulsing insecticide application to offset the timing of natural predator reproductive cycles can lead to more effective biocontrol strategies. But these are, published in really sort of obscure places like chaos solitons and fractals, right? So this is some pretty obscure theory out there. So I wanted to sort of bring a little bit more theory that we're more familiar with into the equation. So the objectives for this first part of my talk is to incorporate temporal variation into some simple resource consumer models, to compare pulsed and continuous predation scenarios. So most of the theory that we have out there assumes predators are constant and continuous. Excuse me. And to contrast these cyclic and stochastic pulse intervals with sort of the classic scenarios for modeling these systems. So I'm going to start off with some pretty classic, simple. With some classic ecological theory. So this is, for those of you not familiar, logistic growth curve for prey. This just simply describes the sort of S-shaped trajectory of a population through time when there's a carrying capacity, so some max number of individuals that can be sustained, which is determined by the saturation point. Now, there's a classic sort of scenario for doing harvest models in which you take this logistic growth and you just subtract from it some amount of individuals you're going to harvest from the population. And this is a commonly used in fisheries, a model commonly used in fisheries, where you can do things like, for example, identify the maximum sustained yield. So how many individuals can you remove from a population and still maximize the growth rate of that population, which turns out to be at half of the carrying capacity. And I'm going to just take that simple harvesting model and make that harvest scenario be pulsatile by using a Dirac delta function here, in which case we just take that harvest total amount of individuals harvested and we break it up into some segments based on that delta function there. So it's a pretty simple scenario for thinking about pulsed predation dynamics, but I think it has some pretty interesting implications. So essentially what I'm going to show you now is a scenario where I simulate the removal of individuals from the population. If the pulses are frequent, i.e. there's small time intervals between pulses, that means the removals are larger. are smaller, and if the pulses are bigger, the removals are bigger. So the total harvest stays constant over all scenarios. And then I compare regularly and stochastically spaced cycles. So just to get familiar with the results, I have 3 different ways of looking at population here. This is population size on the y-axis, and this is the interval between pulses, so large intervals and small intervals, the size of the harvest in each interval, and then the population growth rate. And so we see for the no harvest scenario, population stays at its equilibrium. And if we introduce a continuous harvest using the classic harvest model, where we just subtract h from the population, we see the population size is smaller. So we have a smaller equilibrium population with harvest, and it decreases with harvest rate. So all that makes sense. Now I want to compare that with a scenario where we have cyclic removal. So the same harvest, but removed in small cycles. And we see if we do those removals in a cyclic way instead of in a continuous way, we actually reduce the population size much more than we did in the sort of constant continuous harvest scenario. And if we make those cycles stochastically spaced instead of cyclic, we see an even bigger impact on the population size, where the population size reduces at a faster rate as we increase the interval between pulses and the harvest rate. And the population growth rate is slower. Right? So those gray shaded areas behind these lines also show you that we get sort of 2 possible outcomes with increasing. So it's increasing instability in those populations where they either go extinct or they persist at a lower population size with the pulsed predation scenarios. And that dynamic emerges if we look at sort of what's going on here at a sort of more mechanistic level. So here's a small pulse interval. The blue dot here on each of these graphs is the equilibrium population size with harvest. The green dot is the smallest population size following harvest, and the red dot is the largest population size. So what happens is when the pulse interval is larger, when we convert these to population growth rate scenarios, so now we have population growth rate on the y-axis, we see that with small pulse intervals, the population growth rate is much less variable than when we have a large interval. So we get a huge variation in population growth rates, and that translates into additional instability in the prey population's size over time. So next I wanted to ask, can we use this instability to actually to use pulses as a management tool? So can we actually use this to, say, develop removal strategies for an invasive species or as a biocontrol measure? So I just looked at, in this case, the percent of populations that were eradicated under these different scenarios. So we have the cyclic in green, stochastic in purple, and then solid lines are small pulse intervals, and the dashed lines are large pulse intervals. And you can see that both the cyclic and stochastic scenarios generated a much greater percentage of populations being eradicated than a constant removal rate of the same amount. And so you get this sort of emergent property where these cyclic predation scenarios are actually changing population sizes more than we would expect based on current theory. And we also get, for those scenarios where we do get extinctions, those extinctions happen at a much faster rate. So this is time until eradication, and we see for even lower removal rates, if we do it in a stochastic way, we can cause extinctions at a much higher rate than just doing constant removal scenarios. So just to conclude this first part, this is a little piece of theory that a postdoc and I published last year, or year before last now, that showed pulses and top-down processes can influence prey populations, and that these likely lead to cascades through food webs that affect whole community dynamics in ways that we're not currently capturing with the theory that assumes constant predation through time. And this could have important applications, such as for biocontrol or invasive species management, where often, when we're doing invasive species management, for example, we use pulsed strategies anyway because we get volunteers on weekends to go out and pull things out of the ground or whatever. And if we can leverage that sort of natural pulse dynamic to sort of maximize our eradication probabilities, that might be useful to think about at least and develop this more. Okay. So now I want to move on to the real sort of focus of this talk, which is size dependence and multiple predator effects. And thing I started off with in the beginning, how these things can translate across ecosystem boundaries. Now we know that sort of habitat patches can be spatially structured in terms of trophic dynamics, where you can have top predators that have larger ranges than sort of basal species, and that habitat patches can interact via dispersal through meta-community ecology, right? But one thing that's been less well thought about is that when you have one of the organisms in these habitats that has a complex life cycle, where we have two patches that are now linked obligately through reproduction and metamorphosis of that organism, you can create linked dynamics that are much more strong than those that's sort of expected through dispersal. So to sort of investigate this sort of linked dynamic through complex life cycles, I did some work on red-eyed tree frogs at the Smithsonian Tropical Research Institute in Panama. And this species was particularly good for this question because of their life history strategy. So these guys live up in the sort of forest canopy for most of the year. They come down to the pond during the rainy season from June to December, where they deposit eggs terrestrially on the undersides of leaves that are overhanging the ponds. Those eggs hatch after 4 to 7 days into free-swimming tadpoles, and then metamorphose 1 to 3 months later into little froglets. Now, what made this organism really good for asking these questions is that each one of these stages, because each one of them is in a separate habitat type, actually experiences predation by a different suite of predators. For example, the eggs are attacked by a couple of species of snakes, as well as some wasps. The larvae can be eaten by a variety of invertebrates, such as dragonflies and bellostomatid water bugs. And the metamorphs are often attacked by fishing spiders. Right? So each one of these spiders is interacting with these prey in a quite different way. But one of the really things that made this really interesting for looking at interactions between multiple predators is that you would think that these eggs are really just sort of passive participants in this interaction between the predator and the eggs, between the snake. But it turns out they're not so passive in this interaction. The eggs of these tree frogs can actually detect the vibrational signatures of predators approaching them on the branch, so they can tell the difference between a snake and a wasp and rain and wind. And they actually release this hatching fluid from a gland in the top of their mouths that causes their egg jellies to dissolve. And so they can hatch out prematurely, and up to 40% prematurely. So they can respond to the predator while they might get a mouthful. The rest of these guys will just release this hatching gland and drop into the pond. Right? So you get this behavioral response by the embryos to the predation threat. You can see them dropping out there. Now that has implications, because now what this predator has done is not only reduce the density, in some cases by up to 50% of the eggs around the pond, but it changes the phenotype of the prey. So for example, this is a full-term hatchling, no gills, sort of positively buoyant, so it can float at the water column. These are filter-feeding tadpoles. Versus the one that hatched prematurely at 4 days here, which you can see still has external gills. It's at the surface here, but they actually sink to the bottom and lay there. And they're considerably smaller than this full-term hatchling. Right? And this change in their phenotype from hatching early should change the way that they interact with aquatic stage predators like water bugs and dragonflies. So we should get this sort of interaction between the predators across this ecosystem boundary from land to water. So in order to really sort of talk about multiple predator effects, first we need to ask the question, how do we generate the expectations for how predators are going to interact to know if there's some unique effect of this change in phenotype changing the interaction between predators across boundaries. So the most classic way people have used, and the sort of really common way people have done this sort of null prediction for multiple predator effects, is using something called the multiplicative risk model. Simply, we just look at survival of some prey with predator 1 and the survival with predator 2, multiply them together, and that tells us the expected survival that we should get with both of those predators if they're behaving independently. But if there's some interaction between the predators, or between the predators through the prey by changing their phenotype, then we should get some observed survival with both predators together that differs from that null expectation. Right? So put mathematically, it's just that the expected survival with the two predators is the survival with each predator by itself multiplied together, divided by some control survival. So this, if you do a sort of Google Scholar search for multiplicative risk model, you see this has been used and get like 3,000 citations, right? This is for the last 30 years been the dominant approach. but it's some problems associated with it. Most notably, it conflates predictable nonlinear predator effects with these more difficult to predict emergent effects, like changes in behavior or phenotype that should change the interaction between predators. So what do I mean by that? Well, this model assumes that predation is constant. So what I mean by that is that if we do an experiment, we know how many prey we put in our experiment on the first day of the experiment. We end it 3 days later. We know how many prey are left. We take the average of that and divide it by the number of days, and that tells us the per capita risk per day. Right? In order for that to be true, we have to have two conditions met. One, that means every time a prey is eaten, we're putting it back in. So there's instantaneous renewal of the prey. Or the prey has to have a type 1 functional response, a linear functional response, which shows that per capita risk to prey is independent of prey density. We know for almost all predators this isn't true. Predators have some number of prey where they start to saturate the number of prey they can eat per unit time. And so we describe that kind of scenario with something referred to as the type 2 functional response, that you guys may remember. And this has two important parameters. This attack rate parameter, which describes the initial increase in the number of prey eaten as you increase the number of prey available, and this handling time term, which sets the saturation of that. So when they start spending more time handling the prey they've already captured than they do searching for new prey. Now, if we take this kind of a scenario and we look at per capita predation risk, we get a different scenario, right? In this scenario, per capita predation risk changes from day 1 to day 2 to day 3. The consequence of this is due to something called JENSEN'S inequality, is if we take that traditional approach and assume that predation risk is constant, we're always going to overestimate how dangerous a predator is relative to what it would actually be if we integrated over that nonlinear function, right? This is problematic now because if we take our two predators that we've now estimated using this sort of constant approach and we multiply them together, we're always going to say there's some emergent multiple predator effect. The predators are interacting in some way. So there's bias towards always finding these emergent scenarios that are unpredictable. So just to illustrate that, here I've simulated two predators that are identical, so they're clones of one another, S1 and S2. And if I use the multiplicative risk model to generate expectations of prey survival through time, this is what we would expect with the MRM. However, if we actually look at actual predation over that same time frame, we see that we would always overestimate the mortality rate of prey using the multiplicative risk model, and the duration of the experiment would magnify how big that emergent multiple predator effect is because more and more prey are going to be depleted. Right? This problem gets even worse when you start thinking about now we're not going to have clones of each other, but we're actually going to change the parameters of the attack rate and the handling time. And what we see is that not only is there bias, and this is for additive and substitutive experimental designs, but not only is there bias, the bias is unpredictable. It's not always positive or negative. It could be positive, negative, or neutral, and it's hard to know which one of those you're going to be. So you can't even go back to the literature that's already been published and say which direction the bias is in. So this sort of calls into question, much of what we know about multiple predator effects, because almost all of it is based on the multiplicative risk model. Okay, so second problem is that even if we account for that depletion effect that I just pointed out, most of these models also assume that all individuals of the prey are equally vulnerable to the predator. But I already showed you that these guys are different sizes, for example, and we know that size should affect predation risk in addition to the abundance of prey. So we have the exact same problem here. So if we have prey that are size structured, so we have small, medium, and large prey in our cohort of prey that we put in our experiment, or the prey grow over the course of our experiment, we have the same problem of averaging across that mortality of those prey and assuming that describes average mortality rate. We again will overestimate mortality rate for those prey from a single predator. And again, doing some simulation scenarios, the direction of bias can vary depending on what those parameters are between the two predators. So this is just varying how different they are and which size prey they most prefer, and then how curved the relationship is. And you can again go from positive to negative to positive bias. So it's hard to go back and sort of back calculate from the existing literature whether the bias has been an important factor in our understanding of multiple predator effects. So I needed to overcome this. In order to do that, you need to know simultaneously what is the density-dependent predation risk as well as the size-dependent predation risk for an individual predator. And so the experiments that we did with these red-eyed tree frogs to sort of get around this were to use response surface regression designs. So this is just 7 different levels of tadpole density that we put in our experimental units and 5 different size classes of tadpoles. And each one of these X's would represent sort of a treatment combination. But this would assume that we were interested in all pairwise comparisons of predation risk with these predators. But in this case, all we're interested in is understanding the shape of the function. So we can actually minimize this experiment by focusing on only those treatment combinations that allow us to describe the shape of the surface and not necessarily pairwise relationships. And those included the full regression series of an intermediate size that allows us to get the functional response, and then the full regression series at intermediate density, which allows us to estimate the shape of the size dependence, and then these corners here, which allow us to anchor that surface in space. All right? And so we can also now, once we do this kind of experimental design, we can fit lots of different models to that data and not just assume a specific functional form. So for example, we could fit our 3 common functional response curves, so a type 1, a saturating type 2, or a logistic type 3 for this density-dependent component of this, and we could fit different shapes for the size dependence, and then using a sort of model comparison approach to determine which one of these best captures both the size and density dependence simultaneously. So what that looks like in this particular scenario was a type 2 functional response with the size dependence that followed a power-Ricker function. Whoops. So what that looks like when you fit them simultaneously is that going into the screen here we have increasing tadpole density, so you might be able to see that saturating curve as you go into the screen for the number of eaten, and then tadpole size along this axis where you get that hump-shaped relationship. So now what this allows us to do is go in for any size and density combination of tadpole prey, we could predict their per capita risk of being eaten by that predator. Right? And here's just an example of those data, the actual data curves fit to the data. This is the same exact data points except fitted as a function of prey density on this axis here. So this bottom line here with the big triangles are the largest ones. They weren't eaten because they were in a size refuge. And up here are the smallest ones. And again, as we increase size, we see that hump-shaped curve, highest abundance, lowest abundance. Right? So we can now estimate simultaneously the size and density dependence. Right? But what about when prey grow? Right? In order to really understand the dynamics between this predator and prey, we also have to consider that if we change resource availability, these tadpoles will grow more quickly across the size axis, and that will minimize the amount of time they're in this window of maximum vulnerability. And that should change the predator-prey outcome as well. So we paired this up with a bunch of growth experiments. First, we did an experiment to look at how density of prey affects size, and then how resource availability affects growth rate of those prey. Again, using this sort of response surface regression approach. And I'm just going to quickly show you the data from that. Here's our growth through time, so size of tadpoles through time. Light circles are low densities, dark circles are high densities. And what I'm showing you here in these curves are Gompert's growth functions fit to these data. So this is just standard growth models. And there's just 2 really important parameters in this particular model to focus on. This gamma naught term here, which describes the maximum growth rate, sort of the linear part of these curves. And then this alpha function here, which describes the cost of being large, sort of a metabolic cost parameter. So it describes how these saturate out. And so again, using a model comparison approach, we were able to identify that density was affecting the value of this alpha term here. So the cost of being large. It was more expensive to be large when you're in high density than it was when you're at low density. And so you saturate more quickly. And so we could then model that alpha term as a function of density to actually incorporate that explicitly into our model. Right? Now for the convenience of this is that we can derive from that size function, that Gompert's equation, this relative growth rate or size-specific growth function. And so because this is derived directly from this original equation, all of these parameters have the exact same meaning and values. So the gamma naught here is setting the intercept of this growth curve as a function of initial body size. So now we could actually look at that scenario where I looked at different resource availabilities and measured how that affected growth and model that initial gamma term here, so maximum growth rate, as a function of resource availability. So here we can see this increases in sort of a Michaelis-Menten scenario. And so what that allows us to do then is just basically move this intercept for the relative growth rate to the left or right on this growth rate function based on available resources in the environment. So we can explicitly now capture size and density dependence and resource availability into the growth function. And so using now all this information, we built a model where we can generate a simulation looking at survival of tadpoles through time under a low resource or high resource scenario. And you get some interesting patterns out of that. One is that if you did a short experiment, you would see that those tadpoles in low resources had a higher survival than those in high resources, but that pattern switches as the experiment goes on up to like 30 days when these guys would start metamorphosing. And the reason for that switch is if we look at growth rates. Tadpole size through time here shows that although those individuals are having lower survival initially that are in high resource, they actually enter and exit that window of maximum vulnerability sooner than those tadpoles in low resource environments. They're actually spending 40% more time in that window of maximum predation because of the low resource environment. So this generates some interesting dynamics that you don't capture by just looking at sort of snapshots of predator and prey interactions. But so does our model work? So we now ran a simultaneous cattle tank experiment matched up with our simulation experiments where we simulated the effects of egg stage predators, so 4-day hatchlings and full-term hatchlings, and density, so we did 2 densities, and then we exposed them to either an aquatic stage predator or not. And here are sort of the results from the mesocosm experiment, when neither predator was there as our control, versus the scenarios where we have an egg stage predator. So this is the egg stage predator's effect on body size of the tadpole, on the density of the tadpole, or having both the size and density effect of the predator on the tadpole. And so what we found is that we didn't see really any large costs to hatching early, right? So this is high density, low, small body size. They didn't really survive any worse than the control scenario of having large body size and high density. But we saw a large effect of lower initial densities. They actually had much higher survivals when the initial densities were lower, probably because of a competitive release. And so if we did the boneheaded sort of additive assumption that we might expect, we would say, okay, well then the effects of both would be a slightly positive effect of hatching early and a large effect of having low density, but we don't see that at all. We see something quite different. And we expected that this was a result of that sort of interplay between the size and density dependent growth dynamic and predation risk. So how well does our model do at predicting this pattern? Well, it turns out it does quite well, which sort of confirms this idea that incorporating these nonlinearities can improve our ability to predict dynamics. And note, this isn't sort of immediate short-term dynamics. This is 30 days post-hatching. So we're actually predicting things out quite far in terms of survival, right? Now, to confirm that it was actually the sort of growth component, we did another experiment where we looked at different, now we have the same sort of manipulation of the tadpoles for predator or no predator. And then we have 3 different resource environments, constant density. And in this case, I'm going to show you the results from a dragonfly, but we did both of these with both predators. And again, now we see survival 30 days post-oviposition after the eggs were deposited. In the low, medium, and high resource environment. And we see increasing survival with increasing resource availability, again suggesting that more rapid growth is getting them through that growth window more quickly. And our model, again, captured that dynamic quite well. But what was even more surprising is if we looked at whether or not we could predict the sizes of tadpoles 30 days after hatching, we also, again, did pretty well with our model of actually predicting the sizes of those individuals. And so why this is really important, because this suggests, since these guys are now going through metamorphosis, that we can maybe predict both survival and size at metamorphosis, which are both sort of key correlates of fitness of these kinds of organisms with complex life cycles in terms of long-term reproduction and success, and immediate sort of survival as a metamorph. Okay. So that study really sort of triggered the work that I'm doing currently in multiple predator systems and led me to sort of asking questions. Can we leverage that knowledge to improve our ability to predict how many predators, not just two, are affecting prey and then cascading their effects cascading through ecosystems? And so what we're trying to do in current work that I'm doing in a rock pool system is trying to develop and test a general quantitative metric for defining and predicting predator diversity ecosystem function relationships. And this is sort of merging together, mesocosm experiments, field manipulations and observations, the development of new statistical tools, which I'll not bore you with unless you really want to. I have them at the end. I'll show you all day, but also a little bit of new theory. So now I've been talking about one or two predators, but we actually all are pretty familiar with the fact that most ecosystems are characterized by having multiple predators sharing a single prey. For example, in Yellowstone, it's not just wolves. We also know it's grizzly bears and other things that are changing that dynamic there. So what we really want to be able to do is predict the aggregate effect of many predators on prey and how that cascades down. But the problem is, we're actually, as I just showed you, really bad at even predicting one predator-prey interaction, much less two predators, one prey. So if we get to many predators, one prey, how can we even dream of being able to say anything with any real accuracy? And, this is, an expected problem because prediction is really hard, right? Describing is really easy. Prediction is hard. And if any of you have attempted this, you've run across it before. But for this particular scenario, prediction is hard for a couple of really identifiable reasons. One, there's idiosyncratic methods for describing and quantifying diversity across different systems. So for example, if I go out and describe the diversity using current metrics of my pond ecosystem, it is not comparable to what someone might do in a savanna, right? The metrics are completely idiosyncratic to the system you work in. As I've already pointed out, the models that we use to generate null predictions are ineffective or even biased, so we don't really have anything to compare back to that's really useful. We also, I sort of alluded to this, that empirical and theoretical work is actually being done on different scales. Empirical work is usually short-term snapshot, and then we talk about dynamics, which are things that are at least over multiple generations. And our descriptive approaches, the things that we use to describe what diversity is, and the models we use to predict what diversity effects are actually not well integrated at all. They're completely independent sort of subfields of the same discipline. So just to go quickly through, how do we currently measure diversity? Well, there's species diversity, which many people have already sort of acknowledged is not the best way because species diversity isn't necessarily the thing that matters. What really matters is functional diversity. So what I'm going to focus on here is the ways that we've tried to characterize functional diversity independent of species diversity. So what actually makes them do the things they do in an ecosystem. And one of the more common ones has been variation in body size or variation in traits. So for example, if we have a big and a small and a smaller predator, then we might expect that to be a high diversity system. So these guys might focus on different size prey and therefore complement each other in the ecosystem relative to a bunch of predators that are the same size. So sort of the status quo at the moment in the field is to use phylogenetic distance as a surrogate for functional diversity. In this case, we would expect a community of predators that are distantly related phylogenetically to be more functionally diverse relative to those that are closely related on that same tree. Or the identification of functional traits, and then using some multivariate description of trait space that are linked to function. This might be something like claw size or crushing force or gape size or things like that actually directly have to do with consumptive ability. So in this case, we might expect if each one of these dots represents the traits of a predator, predators that have traits that are spread out from each other, so they're far from the centroid of that distribution of traits, would be more diverse than one where they're all pretty much the same, right? So there's low diversity in that system. So the problem is, in each one of these cases, these are not general ways of describing predator diversity, right? So if we use phylogenetic distance, pairwise distance phylogenetically, that only applies to the phylogeny in this pond and not to this pond, because there's no quantitative relationship between phylogenetic distance and function, right? There's no expectation there. It's a very sort of subjective thing. Or same thing with body size and specific traits. If we use claw strength, and this ecosystem over here doesn't have claws, well, that no longer becomes a comparable metric of functional diversity. So what we've done is try to develop a general theory to predict multiple predator effects on prey, and then use that as a way of also defining functional diversity. So here's our general model for predictive model for predicting diversity effects. So this is just an adaptation of what I just showed you with the red-eyed tree frogs, in that we have, the details of this equation are not important. Just to know that this section right here describes size and density dependent predation for one decay predators eating the same prey simultaneously. So it incorporates size dependence as well. This term right here is prey growth. And this term out here is variation in individual prey growth. Because what we often see is when you have a predation in the environment, you get variation among individuals. And so what this allows us to do now is for any number of predators, describe survival, which is on this axis, through time. And so what you see is the prey are getting killed and growing. This curve starts to flatten out, and we see that the prey size axis is increasing in this direction, and the size of that hill is spreading out, which is accounting for that additional variation in individuals. So this equation basically incorporates simultaneously size dependence, prey growth rate, variation among individuals, and consumption, size-specific consumption risk by any number of predators, and is not subjected to that depletion problem that we started off with. Now this is just for describing a static experimental scenario, but this can also be used to describe dynamics in the same way by incorporating recruitment function for the predators here, which is just the number of prey eaten down here, minus some death rate, and then some recruitment for the prey, which is sort of input rates into this model. So we can actually do this over many generations and sort of see how these dynamics play out, both in short and long time scales using the same model. So then this allows me to go back to the scenario and say, what is function then? Well, for predators, which is what I'm talking about, it's pretty easy to describe function. It's how many prey they eat, right? It's pretty simple. The function of these predators is to eat prey. And so why not then use the parameters of the theoretical models that we use to predict consumption effects long term to describe function, right? We have parameters that we use. Things like attack rate and handling time are parameters that we can measure from any predator, and they have the same meaning for every predator, right? They're not idiosyncratic to system. Attack rate is attack rate, handling time is handling time. The two combined tell you maximum consumption rate of prey. Similarly, we can use our prey size dependence parameters from the equations that describe those to also describe function. For example, we can say which prey do they prefer to eat. This one likes to eat medium prey. Or what's the maximum attack rate? And these are all sort of described by these parameters in the equations that we use to model their dynamics. So if we take those parameters and call them functional traits, and put them back into this trait space, now we have things on this trait space like attack rate on this axis and size dependence, and I'm just putting it in 2 dimensions here for illustration, where now this are different predators. This one has this attack rate and this size dependence. So the centroid now gives us sort of a description of the average consumption rate of this community of predators. The size of the circles incorporates different relative abundances of predators. So large circles have a larger effect on the placement of that centroid. And therefore, if we use the sort of sum of the lengths of these lines, the distances from the centroid as our measure of functional diversity, we can sort of incorporate both density and sort of uniqueness of individuals and their consumption rates as functional metrics. So different predators have different attack rates, handling times, and size preferences. And these things, the functional responses themselves, determine predator function. So let's just use those parameters to link functional traits to the dynamics that we're trying to describe, and we can maybe be more effective at predicting predator diversity effects on ecosystems. And again, this is quantifiable and comparable for any assemblage of predators. It's not idiosyncratic at all because they're parameters to describe their function. So I'm just going to show an example of how we might use this in a three-dimensional trait space. Here we have handling time, attack rate, and size dependence. So I've just added another parameter here. This is the centroid of this community of predators. And let's just say we make these two predators go extinct. Right? Well, if we do that, we can a priori know exactly where the centroid of that community of predators is going to go. And therefore how functional diversity is going to change with the extinction of those two predators. And since all of those dots are still linked to this model, run it through the model, we know exactly how prey consumption is going to change with the loss of those predators. And so we should be able to predict exactly how ecosystem function is going to change, or at least better than what we've done previously. Right? So does it work better than our previous methods? That's the real question. Well, the problem is I could not find a single system where we knew things like body size, phylogenetic relatedness, and identification of various functional traits to contrast them with our approach. So what do? Well, you make up data, right? So I had to make up some data to try to test these things out. And the way I did that was to use some evolutionary models where essentially we randomly generate phylogenetic trees, and then using a Brownian motion of evolution, allow predators with different combinations of those traits, the parameters, to evolve on these phylogenetic trees. And then we can select randomly predators from that simulated evolved predator community and play them out and quantify phylogenetic distance, the traits that they have that sort of evolved independently, as well as the parameters of their functional responses. And then we can make some of those predators go extinct and ask how well can we predict the change in prey consumption given the extinction of some of these predators. So we've kind of just done this in silico experiment. So here are going to be the results, predator diversity metrics for the different scenarios here, morphological, phylogenetic distance, multiple traits, or functional diversity that I'm describing as the functional parameter diversity, and the proportion eaten on the y-axis. And so we see that just looking at some morphological trait, when I do the simulation scenario, provides no predictive power whatsoever in terms of number of prey eaten. And it doesn't get much better for phylogenetic distance or the use of multiple traits, whereas using these functional traits as the parameters, the parameters as the functional traits, actually does an incredibly improved job of predicting the proportion of prey eaten by a given predator community. Now what happens when we make some of those go extinct and predict change? So here's the same plot, except now we have change in diversity after an extinction of 1, 2, 3, or 4 predators. We started off with 6 in this scenario. And the change in the proportion of prey eaten by that predator community after the extinctions. And again, we see a similar kind of result. Not quite as strong, but still considerably better than the alternatives. We're able to still explain 50% of the variation in the change in prey being eaten by our predators relative to things like phylogenetic distance, which is the currently most used approach in the field. Right. How much time do I have? A couple minutes? Okay. So theoretically, at least, it looks like this is working pretty well. But can we test this empirically? Right? That's the real question. And I think that we can, because the approaches that I've proposed for estimating functional responses, getting size and density simultaneously, are short-term experiments that you can extrapolate out to long-term dynamics. And so what we're doing is we're creating communities of varying predator diversity and actually experimentally examining ecosystem function. And we're doing that in this system in Richmond, Virginia. Just to give you a sense, this is right in urban Richmond, Virginia. This is some drone flyovers. You can see downtown here. This is a big granite slab that's been exposed from changing of the river channel. And if you see all these little black dots here on this landscape, each one of those is a little solution pool, rock pool that we have here. And there are actually thousands of them. We have over 800 of them mapped so far. So here's just an aerial view of that system. And in this, we're getting really detailed drone imagery, so we can actually see algal growth, for example, in these pools, and so we're getting some LiDAR data as well where we can maybe monitor ecosystem dynamics. But the real thing that makes this system useful is that there's a single dominant grazer in this system, which is a physid snail, and you can actually see the signature of the presence of large numbers of these snails, and they get really high densities, and they co-occur with 11 different species of predators in these rock pools. And so we can actually now go in and manipulate the predator communities, and there's a single dominant grazer where we can actually maybe test some of the predictions in a simple replicated ecosystem. And then we're pairing that up with laboratory experiments where we're actually estimating the size and functional response relationships for all of those different predator species to see if we can actually predict how different combinations of predators are playing out over short and long-term timescales in the natural system. And this is sort of one of the first results, and it looks like we were able to predict things pretty well in this one experiment, but it's a very preliminary analysis, so stay tuned for the actual real story. And I'll leave you, it was a lot of information, so 3 take-home messages if you don't remember anything else is pulses in the magnitude or timing of predation might have important implications for predator-prey dynamics, and so we should think about that when we're devising our experiments and inferences. For the second part, density, size, and growth are important determinants of the outcome of predator-prey interactions, and failure to consider these nonlinear processes can lead to bias in our expectations for multiple predator interactions. And finally, using the parameters of well-vetted mathematical models as functional traits may improve our ability to predict how changes in predator diversity will affect ecosystems in a more general way. So a bunch of acknowledgments, especially my collaborators and postdocs, and happy to take questions if I have time.
Or tell you about the statistical tool.
I think we have plenty of time for questions.
So this experiment that you talked about, the tree frogs,
Uh-huh.
how did you spooking the eggs? Did you all become snakes on it, or did you just like use your hands to like?
Yeah, so it turns out you can mimic the vibrational signature of a snake by just rubbing your finger over the egg masses, and they hatch.
Something for much more exciting.
No. They did some experiments where they actually put accelerometers in there and measured the vibrational signatures of the different predators and then played those back to actually, determine that they're actually using cues to hatch out. But for the experiments, we went with the more crude approach. Yeah.
How did you classify a functional trait?
So
Did you scale it somehow?
for using the parameters of those models that we have, it doesn't necessarily matter about the scale of it because they only describe, say, instantaneous rates of consumption. And so they're actually comparable no matter what organism or what density, et cetera, that you have. If you fit them through the model, then they should be comparable in that way. So I think that's part of our argument for using those parameters as the functional traits is that they have the same meaning for all predators. And you don't necessarily have to put them in different scales because they're always per predator consumption per time. So it makes them kind of useful in that sense. Does that answer your question?
Do you assume that your instantaneous rate of consumption is consistent in like a simple predator community and a complex one? Are you assuming no interference between predators?
So, very good question. This is the null model that I'm proposing here. Right? And so basically what we're trying to say is lots of the things that we've attributed to things like interference or changes in prey behavior or prey phenotype have actually potentially been biased in the null models that we've been using. And so by incorporating these known nonlinear processes, our null model encapsulates a lot more of those things so that we can actually quantify true emergent effects or interference dynamics. So these are assuming independent interactions between predators. And so actually one of the experiments that we've done, the good thing about the system is the snails show strong behavioral responses to many of these predators. You put in a crayfish and they climb to the top of the water. You put in a fish, they hide under a rock. So you can actually get really strong. They also change the morphology of their shells. So we know we have predator-induced phenotypes in the snails. And we are doing simultaneously, we've created agent-based models that duplicate these deterministic sort of models that we had, these partial differential equation models. And we're thinking between the two, we might be able to ask, can we actually quantify how much of the outcome is due to sort of trait-mediated interactions, or whatever you might want to call them these days, non-consumptive effects, by comparing the agent-based model to our other models. Yeah, that's a good question. I meant to say that, so if I didn't. These are null models. Better null models. Yeah. Other questions? Yeah.
If you were considering like a social predator, would you be able to think of like the group of individuals as one single unit when you're thinking of like handling time and attack rates, and apply like the functional result that way, or would you have to do it like on per individual basis though?
So I think, again, if we use this as the null model, like those, the sizes of those circles in our functional trait space represents abundance of the predator. And if we got some outcome that was, say, higher mortality than we predicted with the model, we might be able to attribute that to facilitative hunting behavior, like, or the inverse of interference. Another thing that we've talked about doing with this is, there's a lot of work out there right now on individual personality. So you could actually populate that entire trait space with different individual predators potentially, and actually ask how important is variation among individuals for driving prey dynamics. So we've thought about how to incorporate more of those things, but this is as far as we've gotten so far.
Do you have plans to expand this to multiple herbivore communities? Because it's nice to have multiple predators. That's definitely more realistic, but having multiple herbivores is more realistic still.
We haven't considered expanding it beyond this yet. I mean, other than hopefully it works well in our system. And then if it does, then we can start thinking about generalizing it to other scenarios. I don't want to say too much yet because we haven't even tested it in our very simple food webs. So I hope so.
And a better question. Can you account for a predator that during part of its life cycle is not a predator? Like something that as a larva or as a nymph or something is some sort of herbivore, and then later on goes on to be like a big bad predator? Happens a lot in insects.
Yeah, so like temporal variation in the timing of predation is not something that we've done at this point. We've kind of assumed all the predators are in the environment at the same time. So one of the things by modeling it after this rock pool system is that a lot of the predators are input basically by flooding events. And so you can get these predators that are input to the pools and the snails, and then the water goes down and they're kind of stuck there. And so that's sort of the idea we're working off with. But obviously, as I pointed out in the first thing, the pulsed nature or the stochastic dynamics could be really important. So one of the things that sort of has that characteristic in our system are dragonfly nymphs. So a dragonfly comes into one of those rock pools and oviposits. You basically put this murderous community of like these hyper-aggressive predators that then will metamorphose and leave, and then you've left the pool behind again. So that's a place where you get these kind of pulsed predation events that we're going to try to explore, but haven't yet.
I got another, I don't want to Bogart. No. I'm curious about, there was your figure where you explained the predictive power of prey size. Most of them were really predictive. It was predictive across most of the prey size classes, and then there was one that wasn't, and I was really curious about it.
Yeah, so our best guess was actually two scenarios where we underestimate size.
I don't want to drag everyone along.
No, it's okay. There's two cases, and I think both of those cases are where I think the thing that our model is missing is endogenous production from the individuals themselves, and so that's our best guess.
It was like the middle size class or something where the red dots are really far apart.
Yeah, and we don't have a good answer for why we missed those, but qualitatively it was the same pattern. But yeah, quantitatively they didn't overlap the confidence intervals in those two scenarios, and we haven't got a good explanation for that yet. No, that's a good question.
When you talk about the specifically handling time of predators handling prey, does you also include in that definition things that are kind of inject venom and then sit and wait for the thing to die?
Yeah, so I mean, for something like a venomous snake or spider, yeah, I assume those would be really long handling times. So the search time would all go into the attack rate term. Then everything after capture and then finishing consumption goes into the handling time estimate. And so those would be things, a lot of sit-and-wait predators probably fit that category. Would have really long handling times, or long time periods between prey events.
But that's assuming that the predator could not say, essentially, like, oh, I'm waiting for this guy to die, I'm going to jump over to this other plant.
Yeah, so there are some scenarios where a little bit harder to wrap into that framework, and I think you could do it with a little effort. Partial consumption is another one that's hard to do a handling time for, because, I'm eating, I'm going to, cats do this, I'm going to go eat this one now, because I only ate half of that one. So I think those things people have worked on that, and there are ways to incorporate that additional complexity. And we haven't built it into the models, but I think it's possible to do so. Or those may be special cases where exceptions that it doesn't apply. Yeah. Micropredators are another one that would be really hard to do in this framework. And we've also worked really, really hard to incorporate parasites, but that's also weird because they have this sort of flip dynamic where, it's the number of parasites per prey, not the number of prey per predator. So we haven't successfully done that yet either.
Do you want to show us your statistics?
Oh, it's pretty boring, but
We could take a break and then do it for those that are interested.
it's real quick. It's not actually a lot there. So one of the things that we've been criticized for in the past is when you want to do. Let me see if I can get this to. Whoops, that's not what I meant to do. Still there. Good. Is these response surfaces that we propose that you use, right? If I can get to one.
Let's thank Dr. McCoy while he's opening his thing. People are dribbling out. I'm going to hang around.
So,
and look at
with
statistics,
fitting these kinds of things, right? This, people say this is really hard, and the other thing you can't do with this is you can never run enough experiments to do lots of replicates of this. So you have to have things temporally blocked or spatially blocked in some way. And so all of our statistical approaches is sort of the first rendition of being able to fit these nonlinear three-dimensional functional responses with random effects. So if I can get back to the end of my talk. So basically what we've done is we've adapted 80 model builder using Rogers' random predator equation, which accounts for the depletion effect. And we can incorporate size dependence to actually simultaneously, so you run it in R. It's using template model builder. Then it calls in a C++ algorithm that allows us to sort of estimate both the random effect and the fixed effects, or the parameter estimates from the curves. And it's super fast. That's the big thing. So it's just an example of the coding that goes into it. But the boring part is that we actually show that we have pretty small bias on our fixed effects parameters, and we got good coverage. So the statistical models are working, for those of you who think about these things. Not as good for the random effect, but this is true for almost every random effects model. If you guys aren't aware of that, they're all biased in terms of the estimate of the random effects for the most part. So we're in the range of sort of linear random effects models as well in terms of the bias and the estimate of the random effect. And we're not as concerned about it because our coverage is still quite good. Right? We get the answer we expect most of the time. So that's all the statistic stuff is. We developed a new R library for fitting functional responses with random effects and size dependence. So if you want to do this work, you can now actually analyze your data.
What's the package called?
Well, it's not actually available yet, but it's called functional response at the moment, but it'll probably have a better name when we put it actually on CRAN.
Okay.
But if you want it, you can contact me. I'll give you the GitHub for it. Yeah. In the coverages? I'm not sure what's generating that asymmetry in coverage. we've been exploring that a little bit, and this is with my collaborators at McMaster University. So, I don't know right off the top of my head. But. Thanks, everybody. Go get beer.